Today in lab we'll be studying Exercise 40, Anatomy of the Urinary System, and Exercise 41, Urinalysis. Metabolism of nutrients produces wastes that must be eliminated from the body. The urinary system is primarily responsible for the removal of nitrogenous wastes from the body. In addition to this excretory function, the kidney maintains the electrolyte, acid-base, and fluid balances of the blood, and is thus a major homeostatic organ of the body. Malfunction of the urinary system, particularly of the kidneys, leads to failure in homeostasis, which, unless corrected, is fatal. The urinary system consists of the paired kidneys, paired ureters, single urinary bladder, and single urethra. Urine is transported from the kidneys to the bladder via the ureters. Urine collects in the bladder, where it is temporarily stored until it is excreted through the urethra. Notice that the paired kidneys are not positioned at exactly the same level. Because it is crowded by the liver, the right kidney is slightly lower than the left. The renal arteries diverge from the descending aorta to supply the kidneys. The renal veins drain the kidneys, while two ureters carry urine from the kidneys, moving it by peristalsis to the bladder for temporary storage. Within the urinary bladder, we can see the point of entry of the two ureters into this organ. The single urethra drains the bladder. The triangular region of the bladder, delineated by the two openings of the ureters and the single urethra, is referred to as the trigone. In the male, the urethra is approximately 8 inches long, travels the length of the penis, and opens at its tip. The male urethra has a dual function. It carries urine to the body's exterior, and it provides a passageway for semen ejaculation. Thus, in the male, the urethra is part of both the urinary and reproductive systems. In females, the urethra is short, approximately 1.5 inches long, and serves only to transport urine to the body's exterior. In lab, we will be performing a simple kidney dissection and identifying the basic anatomical structures of the kidney, such as the renal artery and vein, renal pelvis, major and minor calyces, renal capsule, renal cortex, renal medulla, renal pyramid, and renal column. Blood flow reaches the kidneys via the renal arteries. Arteries branch several times before reaching the nephrons, the functional units of the kidney. Each kidney contains over 1 million nephrons responsible for filtering the blood and forming urine. There are two types of nephrons, cortical and juxtamedullary. Cortical nephrons are most numerous, making up about 85% of nephrons. As their name suggests, they're located almost entirely within the renal cortex. Juxtamedullary nephrons are located deep in the cortex, with long nephron loops that penetrate into the medulla. Each nephron consists of two major structures, the renal corpuscle and the renal tubule. The renal corpuscle consists of the glomerulus, which is essentially a ball of capillaries, and the glomerular capsule, also known as the Bowman's capsule. The main function of the renal corpuscle is glomerular filtration of blood to produce a filtrate that continues through the nephron. The renal tubule consists of the following: the proximal convoluted tubule, or PCT, the nephron loop, also known as the loop of Henle, with a descending limb and an ascending limb, the distal convoluted tubule, or DCT, and the collecting duct. The main functions of the renal tubule are reabsorption of urinary constituents back into the bloodstream, and secretion of additional urinary constituents via the peritubular capillaries. Urine formation is the result of 3 processes: filtration, reabsorption, and secretion. To discuss these 3 steps, we will use this simplified schematic of an uncoiled nephron, allowing us to trace the path of blood flow through the renal microcirculation. Note that the afferent arteriole feeding the glomerulus is larger in diameter than the efferent arteriole draining the bed. This creates a high hydrostatic pressure that forces fluids and blood components smaller than proteins out of the glomerulus and into the glomerular capsule, forming the filtrate that then continues through the nephron. Blood cells and plasma proteins are too large to pass through the glomerulus into the glomerular capsule, and therefore should not be found in urine. As the filtrate moves through the renal tubule, water, glucose, and amino acids are reabsorbed and returned to the blood via osmosis and selective active transport. Ions are also reabsorbed based on the body's current needs to maintain pH and electrolyte composition. 75 to 80% of reabsorption occurs in the proximal convoluted tubule. During tubular secretion, substances such as hydrogen, potassium ions, and creatinine move from the peritubular capillaries into the filtrate to be disposed of in the urine. Let's review the three processes that result in the formation of urine. First, glomerular filtration forces fluids and small blood components through the nephron as filtrate. Larger blood cells and plasma proteins are kept out of the urine. Next, during tubular reabsorption, water, glucose, amino acids, and certain ions are reabsorbed and returned to the blood. Glucose and large amounts of amino acids should not be present in urine. Finally, during tubular secretion, additional substances move into the filtrate to be disposed of in the urine. This leads us to exercise 41, where we'll be performing a urinalysis to analyze urine samples in lab. Based on what we know about filtration, reabsorption, and secretion within the nephron, we can determine normal urinary constituents, as well as abnormal urinary constituents that may indicate an underlying health condition. Water is the largest component of urine, accounting for 95% of its volume. The second largest component of urine is urea. Nitrogenous wastes in urine include urea, uric acid, and creatinine. Other normal constituents of urine include sodium, potassium, phosphate, sulfate ions, and small amounts of calcium, magnesium, and bicarbonate ions. Abnormal urinary constituents include glucose, plasma proteins like albumin, ketones, red blood cells, white blood cells, hemoglobin, nitrates, bile pigments, and casts. The presence of abnormal urinary constituents can point to possible health conditions. The table in your lab manual summarizes clinical terms and possible conditions linked with high levels of abnormal urinary constituents. For example, high levels of glucose present in urine may indicate uncontrolled diabetes. Before coming to lab, be sure to read through exercises 40 and 41. Take your online Canvas quiz the day before your scheduled lab, and as always, reach out to your lab professor with any questions or concerns.